Hi, today we'll talk about signal transduction. As you can see, signal transduction can be defined as the transformation of the signal from the cell membrane until reach into the nucleus through successive chemical reactions that involves a lot of proteins. There are intercellular signals which are interpreted by complex machinery in the responding cell that allow each cell to determine its position and the specialized role in the body to ensure that each cell divides only when its neighbor cell dictates that. The importance of such social controls on cell division becomes clear when the controls fails that result in a cancer, which usually kills the multicellular organism. An animal cell contains a complex system of proteins that enables the cell to respond to signals from other cells. The system includes cell surface and the intracellular receptor proteins, protein kinases, protein phosphatases, and GTP binding proteins. Signal amplification commonly occurs downstream from cell surface receptors. So you can see here, once a signal like epinephrine binds to one GPCR molecule, this leads to amplification through the generation of adenylyl cyclase. These adenylyl cyclase, another round of replication, lead to the formation of a cyclic AMP, which is the first level of amplification. This activates protein kinase A, in which each molecule of protein kinase A will activate a set of enzymes, and this is considered the second by the help of 4 cyclic AMP molecules will activate 2 pKa, and this is the second level of amplification. For the third level of amplification, each enzyme will activate or do its job on the product, and this is the third level of amplification. Extracellular signaling molecules are recognized by specific receptor on or in target cells. Cells communicate by means of hundreds of kinds of signaling molecules, includes a lot of things including growth factors, amino acids, retinoids, proteins, etc. Most of these signaling molecules are secreted from the signaling cells by exocytosis. Others are released by diffusion through the plasma membrane. So you can see here that the molecule have been secreted by exocytosis to reach a receptor on a target cell. But in this case, some receptors remain tight, and the signal molecules are tied to the cell surface and only influence the cell in contact or just nearby neighbor cells, and this signaling by plasma membrane bound molecules. Regardless of the nature of the signal, either secreted or bound to the surface of the membrane, the target cell responds by means of a specific protein called a receptor. A receptor. It is specifically binds a signaling molecule and initiates a response in a target cell. Mostly transmembrane protein. They are mostly transmembrane protein on the target of the cell surface. When they bind to an external signaling molecule, a ligand, they become activated. Activated receptor generate a cascade of intracellular signal that alter the behavior of the cell. So you can see here the hydrophilic signal molecule is binding to the cell surface receptor on the plasma cell membrane. In some cases, the receptors are inside the target cell, and the signaling ligand has to pass and enter the cell to activate this receptor. This signaling molecule therefore must be small and hydrophobic to diffuse across the cell membrane. And as you can see in the figure, the signal molecule have been carried by a carrier protein until reaching the cell, and it can diffuse because it's hydrophobic, as we said, to the intracellular receptor. Secreted molecules mediate 3 forms of signaling: either paracrine signaling, synaptic signaling, or endocrine signaling. The paracrine signaling, you can see that the signal coming from the cells, from the cell, only affects cells nearby, so it's like a local mediator to the target cells. While in synaptic signaling, which involves neurons, nerve cell is involved through its axons. It mediates, it conveys the neurotransmitter to the target cell. While in the endocrine signaling, the target cell, which is the endocrine cell, releases its hormone into bloodstream that circulate through the body until reaching its target cell. So the endocrine signaling is slow because there is a diffusion and there is a blood flow. Nerve cell can transmit information by electrical impulse reach 100 meters per second. Once a neurotransmitter released from a nerve terminal, it has to diffuse less than 100 nanometer to the target cells, a process that takes less than a millisecond. Hormones in the endocrine signal are greatly diluted in the blood. Neurotransmitters as well are diluted but are much less dilution and can achieve local high concentrations. Neurotransmitter receptors have a relatively low affinity for their ligand, which means that the neurotransmitter can dissociate rapidly from the receptor to terminate a response. And you can see here the difference between the synaptic signaling, the difference between the synaptic signaling and the endocrine signaling. You can see that various neurons can convey the signal to different target cells based on the axon's terminal. While the endocrine signaling have the various endocrine cells release their different hormones, and each hormone will bind to a specific target cell after passing in the bloodstream based on the receptor of the target cell. Autocrine signaling can coordinate decision by a group of identical cells. Cells can send signal to themselves or other cells of the same type. During development, for example, autocrine signaling may be used to encourage a group of identical cells to make the same developmental decisions. You can see here that the signaling cell releases a signal to the neighboring cells, and these signaling cells, this mode of signaling is called autocrine. We have a group of identical cells that target cell receive this signal lead to amplification, since a lot of signal have been secreted from the neighbor cell. The gap junction as well allows signaling information to be shared between neighboring cells. The gap junction is a specialized cell-cell junction that can form between closely opposed plasma membrane, connecting the cytoplasm of the joint cells via a narrow water-filled channel, as you can see from the figure. This allows the exchange of small signaling molecules such as calcium, cyclic AMP, but not macromolecules such as proteins and nucleic acid. Different cells can respond differently to the same chemical signal. A cell reactor to its environment varies according to two conditions. First, the set of receptor protein that the cell has on its surface. Second, the intracellular machinery for interpreting the information that it leaves. The perfect example of that is the acetylcholine example. The acetylcholine, it's a neurotransmitter. You can see that once it's been recognized by a skeletal muscle, it leads to the contraction of the skeletal muscle. While the same acetylcholine, once recognized by a cardiac muscle, heart muscle, it leads to the relaxation. While the same as well acetylcholine recognized by a secretory cell, it leads to secretion. So now we have the same signaling molecule, but we have different receptor and different target cell and different effect. There are three known classes of cell surface receptor proteins: ion channel linked, G protein linked, and enzyme linked. For the ion channel linked receptor, you can see this ion channel is not operated until the ligand is bound to it, and this leads to opening of the channel and the internalization or influx of the ions. While for the G protein linked receptor, you can see it's once the receptor is bound to the ligand, the G protein got activated. The activated G protein will activate another enzyme, and this will lead to the opening of the ion channel. While in the third case, the enzyme-linked receptor, the ligand actually it's by its own self it have a catalytic domain activity that lead to the activation, since it have enzymatic nature after the bound to the ligand. Activated cell surface receptor trigger phosphate group addition to a network of intracellular proteins. So the signal binds to the receptor, and this leads to the signal until reaching the nucleus, which lead to activation of genes that change the behavior of the cell. This intracellular signaling protein, in which proteins that is phosphorylated by a protein kinase and the protein that are induced to GTP when the signal arrives. So there is two modes of conveying the signal through this protein until reaching the nucleus. The first one by the help of phosphorylation of a protein kinase. The second one by the it involves GTP. So you can see here in the first figure, you can see that once the signal is in, this leads to the activation by kinase by the use of the ATP. This leads to addition of a phosphate group on the substrate, and then the signal is conveyed. While in the other case, when the signal is in, GDP is changed for GTP, and hence the signal is conveyed to the other molecule, and then the signal is moved on through the cell. Most of the kinases are serine, threonine, or tyrosine kinases. Survivor or proliferation. These are a complex cell behavior stimulated by a specific combination of signals rather than by a single signal. The cell has to combine the information coming from separate signals so as to make a decision, a response to live or die, or to proliferate or stay quiescent. This equivalent to a microprocessor in a computer. So you can see here in the first figure, you can see in order to decide what the cell do, it needs a signal from two different receptors by two different molecules signals. This leads to the activation of the protein Y by the help of two activation signal coming through a successive reactions of intracellular protein until lead to its activation. Another mode of conveying the signal is as well coming from multiple receptors with different signaling molecules, lead to two different protein subunits. Each of them is activated by its own receptor, and then the binding of these two subunits, these two activated subunits, will lead to conveying of the signal downstream of the initial signal. G-protein coupled receptors. So we'll see here how the G-protein operates. The G-protein coupled receptor, it's a transmembrane protein. You can see that it got activated once a signal or a ligand bind to it. The active receptor, once the receptor, the binding of hormone, this induces a conformational change in the receptor. This leads to the binding of the G protein, which is composed of three subunits, alpha and beta and gamma. This activated receptor binds to the alpha subunit of the G protein. The binding induced conformational change that leads to the in G alpha leads to the displacement of the GDP by GTP, and the alpha subunit will dissociate from the G protein and will bind to the active effector molecule on the cell surface. This hormone dissociation from receptor and binding to the active effector will convey the signal from the receptor protein to the effector molecule. The signal is relieved. When the signal is removed, this leads to the hydrolysis of GTP of the G alpha subunit to be a GTP, and hence the signal is terminated. So as an example, one for that is the cardiac muscarinic acetylcholine receptor that activates a G protein that opens a potassium channel. So you can see once the acetylcholine binds to the receptor, it's the muscarinic acetylcholine receptor, the recruitment of the G alpha subunit to this receptor by the displacement of GDB to GTB, and the binding of the gamma and beta subunit of the G protein to the closed potassium channel will leading to the opening of the channel and the efflux of the potassium to the outside, leaving the membrane with a hyperpolarized state. So the acetylcholine binds to the nicotinic acetylcholine receptor in case of striated muscles, lead to its contraction, while when it's bind to the muscarinic acetylcholine receptor in the cardiac muscles, this lead to its relaxation. So you can see here different effect based on the type of the receptor and the type of the target cells. As a second example, GPCR activation of IP3 diacylglycerol pathway through phospholipase C PLC triggers the release of calcium from the endoplasmic reticulum. So you can see once the receptor is activated by the binding of the ligand, this leads to the conformational change of the G protein, leading to the activation of the phospholipase C. This phospholipase C will break down the PIP2 will break it to IP3 and the DAG diacylglycerol. The diacylglycerol stayed in the membrane while the IP3 will migrate to the IP3 gated calcium channel. Once the IP3 bound to the calcium channel on the endoplasmic reticulum, step number 2 and 3, the calcium is released from the stores in the endoplasmic reticulum into the cytosol. Once released, this calcium will bind to and activate the PKC that after that is bound to the diacylglycerol, leading to the conveying of the signal to the cytoplasm through the phosphorylation of a number of substrate. As well, you can see that the calcium can be internalized through store-operated TRP calcium channel. And this mechanism is used when the calcium is depleted inside the endoplasmic reticulum. For example 3, the membrane-localized Tubby transcription factor is released by activation of phospholipase C. You can see start initially with the same mechanism activation of the receptor and the conformational change of the G protein leading to activation of phospholipase C. The phospholipase C, the Tubby protein, initially is bound to BIP2. Under the effect of phospholipase C, it will break down BIP2 into DAG, diacylglycerol, and IP3. The breakdown of BIP2 will release Tubby into the cytoplasm. The Tubby is characterized by having two major domains, a DNA binding domain, which enables this protein to bind to the DNA, and have as well transcriptional activation domain that enables this protein to initiate transcription of specific genes. So once the Tubby protein is liberated from its binding with BIP2, it will be recruited into the nucleus and bound on the DNA and initiate a transcription. This Tubby gene actually expressed primarily in brain and have been involved in control of eating behavior and as well involved in obesity, and they found that the muted Tubby gene in mice develop obesity. Growth factors. So growth factor actually it's a type of the signaling molecules. Definition and facts. So growth factors are polypeptides that regulate the growth of both normal and pathologic cells. Unlike classic hormones, the action of growth factors are not restricted to specific tissues, but may function in diverse organs. GF growth factors have specific receptors. They may come from both malignant or normal tissues. The protein product of oncogenes include growth factors or growth factor receptors or signaling molecules. The normal function of the growth factor. What tissue have their growth regulated by growth factor? Basically, pretty much all of the tissues is regulated by growth factors. What cellular process respond to growth factors? Basically, the energy use, cell shape, motility, most of the functions. How do cells of different tissues coordinate their actions? Both growth factors crosstalk and cell-cell contacts. So you can see here a simplified model. You can see a growth factor bind to the receptor is phosphorylated. This leads to a signaling cascade through the phosphorylation or activation of a signal of a successive protein, leading to the activation of effector protein, which either have an extranuclear function or have an intranuclear function. Once internalized into the nucleus, it will lead to a gene expression in the target protein. So scientists have used these three levels for three therapeutic targets in case of diseases. We will talk now about the general characteristics of growth factor signaling. Signal transduction by growth factor receptor tyrosine kinases is a multi-step process. Ligand binding induced dimerization of growth factor receptor tyrosine kinase. This tyrosine kinase activation and phosphorylation leads to the phosphorylation of a tyrosine residue on the receptor. This leads to the recruitment of signaling intermediate proteins to the growth factor receptor tyrosine kinase. The transmission of signal by activating this leads to transmission of signal by activating cytoplasmic signaling pathway. So we can see here, this is a cell membrane separating between the exterior and the interior of the cell. And here is, for example, an insulin receptor, which is an example of a tyrosine kinase receptor. Once the signal of the insulin comes in the contact of the receptor, a dimerization occurs, and hence the signal bound to dimerized receptor. And soon after that, since it has a tyrosine activity, it leads to the autophosphorylation of a tyrosine residue on the receptor. This phosphorylation on the receptor will lead to the recruitment of intracellular protein like IRSR1, insulin receptor substrate 1, that binds to the phosphorylated receptor. What is the major signaling pathway? One of the major signaling pathways is insulin signaling. The major advance in understanding cell growth and proliferation have come from the discovery of the intimate relationship between three major pathways: the PI3 kinase pathway, the mTOR mammalian target of a rapamycin pathway, and MAP kinase pathway. For the PI3 kinases, phosphoinositide 3 kinases, this is the best understood, important constituents of critical signaling pathway, key player in signaling intracellular signaling network, engaged by many growth and survival factors which regulate cell proliferation, growth, survival, and apoptosis. Actually, the PI3 kinase as a protein, it consists of two subunits. A P110 catalytic subunit bound to a regulatory subunit called the P85. The P85 contains a domain which is called the SH2 domain that can associate with tyrosine phosphorylated protein that initiate the activation of PI3 kinase. In other words, once the receptor is phosphorylated on a tyrosine, the P85 of the PI3 kinase have a SH2 domain. The characteristic feature of SH2 domain is only bind with a phosphorylated tyrosine, and since the receptor is being phosphorylated on the tyrosine residue, so the PI3 kinase is recruited to bind to the receptor once it is activated. Members of PI3 kinase family can also be considered as an oncogene because they control cell cycle progression, cell proliferation differentiation, survival, vision, and metastasis, and angiogenesis. So any alteration in these in the PI3 kinase may lead to cancer. Looking into the signaling pathway, you can see that the insulin, once bind to the receptor and dimerization, autophosphorylation occurs for the receptor. This leads to the recruitment of the IRS1. This IRS1, with the binding with the PI3 kinase, which is a kinase, this kinase will lead to the phosphorylation of a PIP2 into PIP3. This, of course, can be antagonized by another protein, which is a phosphatase, which is called P10. The phosphorylation and the formation of a PIP3 phosphoinositide triphosphate lead to the recruitment of BDK1 and which activate PKP, but still the signal is on the surface membrane of the cell. The BKP, although it is activated by BDK1, but is still in the inactive state and need another signal for activation. This BKB, once be released in the cytoplasm, it's now active. This BKB will inhibit a complex called the TSC1, TSC2. This complex actually aids in the formation of REB-GDB from REB-GTP. And REB-GTP is an important upstream effector of an important complex called the mTOR1 complex. This mTOR complex 1, once is activated by REB-GTP, it leads to, this is actually a kinase as well, mTOR. This leads to the activation of another kinase which is called S6K kinase. This S6 kinase, to be fully active, it needs another signal from BDK1. So once it is fully active and got the 2 signal, it phosphorylate a 40S ribosomal subunit. This lead to the initiation of the protein synthesis and the control cell growth and viability. So you can see this is a signaling pathway that leads by the end to the formation, to the starting of the machinery of the protein synthesis. This signal can be antagonized by a number of ways, like the use of the rapamycin, which is a compound used to block the mTOR activity, so block translation. It is a strategy used in cancer treatment by enhancement of PTEN, the phosphatase, to stop the further activation of BTK1 and of course after that S6K, and by another inhibitor which is called the Wartman or LY294,002. These two kinds of inhibitor can inhibit BI3 kinase. So these you can see that there is 3 methods to block this signaling pathway. One of them is naturally occurring, it's a PTEN, and the other two actually it's a 2 compound can be injected into the cell to block the signaling pathway. As well, they found that S6K itself can inhibit IRS1, and this is a kind of a negative feedback. Once S6K have finished by its function, by another mechanism it can inhibit the initiation of the signal from the top. The rapamycin actually it's a natural antifungal activity, have a natural antifungal activity. It discovered late in 1970, and it is a potential anti-cancer agent from mid-90s, and it enters clinical trials in 2008. So, as we said in the last slide, S6K to be active, it needs 2 signals, one from the BTK1 from the surface membrane and from mTORC1. These 2 signals activate S6 kinase. Of course, BTK1 is activated by PI3 kinase, and mTORC1 have been found to control cell size and proliferation. Once S6K is activated, it controls a lot of other processes like inhibition of IRS1, which lead to insulin sensitivity, inhibition of bad protein, which inhibits cell survival, inhibition of eukaryotic elongation factor 2K, that inhibit eukaryotic elongation factor 2, and so it promote protein synthesis. It have the effect as well in controlling the cell size, the glucose homeostasis, the regulation of stability of HDM2 protein, and the regulation and the activation of CREM, which is a transcription factor. As well, a lot of researchers have not been proven full proof yet that S6K is involved in DNA in RNA decay. So looking closely into S6 kinase, we can see that it have 5 different domains: a non-catalytic amino terminal domain, and the regulatory N-terminal domain, and the centrally located kinase domain, and the regulatory kinase extension domain, and the autoinhibitory pseudosubstrate domain AI, as you can see from the figure. And you can see on the sketch of the protein of the S6 protein, this is a diagrammatic sketch of the different domains of the S6 kinase. You can see the sites of the phosphorylation on the tyrosine residue from the BTK1 and the mTORC1. If we look closely for the activation of the S6 protein, you can see that it is composed of 502 amino acid, this protein. And you can see initially it is in the active state, as you can see the S6 kinase in the active state. Once there is a calcium binding, it leads to the opening of the protein. With further STP kinase phosphorylation, this will lead to further opening of the kinase. With the activation of mTORC1, as we said earlier in the last slide, this leads to further opening of the kinase, but is still in inactive state. Once there is the other signal from BTK1 comes on the other threonine residue, you can see it's fully active. And scientists have managed to see the structure of this protein under rotary, a kind of electron microscope called the rotary shadow electron microscope. This reveals the inactive and active forms of the protein. So you can see this is inactive, the real image under the rotary shadow electron microscope, and this is the intermediate stage, and this is the fully active form of the protein. This rotary shadow electron microscope is a very high resolution microscope that show a lot of different structure and have been used widely. And this image show how the potential of this rotary shape microscope, and it can grasp a lot of details in the structure they use to photograph. Thank you.